So welcome students, this is Dr. Balding and today we're going to be talking about microbial genetics. So we have finished our discussion of microbial metabolism. We discussed the metabolic pathways that are found inside the cell, both catabolic and anabolic, and looked at some specific pathways that are major ways in which biomolecules are either built or broken down. And so you guys know that there are enzymes which are mostly proteins, intermediates and products in the cells. How these pathways come to exist is a matter of genetics or the study of heredity. And that includes the study of DNA as the genomic material or the material of heredity for most organisms, including prokaryotes, but will also include RNA because some viruses have RNA. So when we get to the virus section, we'll talk about RNA genomes. So much of what we know about genetics has been elucidated in microorganisms. So I'm going to go through a few just definitions to start off here. Genetics, of course, is the study of inheritance and heritable traits. In here, or in this section, we're going to talk about genes. Genes are the single functional units of DNA, usually determines a single characteristic of a cell or encodes for a single protein or subunit of a protein. Genes are found in places called loci. The singular for that is locus. So that's the region where a gene is located. And if you're looking at an organism that has more than one copy of a chromosome, like we do, so bacteria are what we call haploid. So they will have one copy of their chromosome and one allele for a particular trait, okay, that's located at one locus. If you're looking at an organism that has duplicate chromosomes like we do, or diploid organisms, humans will have two alleles for each gene, or basically they're different versions of that gene at the same locus. So usually two in humans, one in bacteria. All right, so another idea that we'll talk about here is the genome. The genome is the totality of genetic material in the organism. It contains all the information for the structure and the functioning of the organism itself. All right, so let's move on. So we're going to quickly review nucleic acids. We covered this in our chemistry section, but we'll do another quick review here, maybe include a little more information. So you guys know that the nucleic or the genomic material of an organism is going to be made up of nucleic acids. And in, like I said, most organisms that's going to be DNA. But in viruses, we're going to be talking about RNA genomes. Nucleic acids are one of your biomolecules that are made up of monomers called nucleotides. Nucleotides, of course, have several different components. They have a nitrogenous base, right? They have a sugar, a 5-carbon sugar that's either a ribose sugar or a deoxyribose sugar if we're talking about DNA versus RNA. And then it has a phosphate group. Now, when in DNA, these and in RNA, when these nucleotides pair, okay, or which they can, they're called base pairs. So here, if you look on this particular diagram, we've got a base pairing here of A and T. And you guys know adenine pairs with thymine. Guanosine pairs with cytosine. So it's always a purine, a double ring structure, pairing with a single ringed nitrogenous base, right, a pyrimidine. And they're going to hydrogen bond, right? So this hydrogen bonding occurs across the helix in a DNA molecule, and you'll notice that these base pairs, right, have hydrogen bonds that differ in number. AT pairs, so this one and this one, have 2 hydrogen bonds. Cytosine and guanine, right, make 3 base pairs. These hydrogen bonds are weak individually, but when you have many, many of them, as we do down the center of this, these paired strands of the DNA molecule, they can be quite strong. Now, why do I point out the number of them? Later on, we're going to talk about mechanisms of regulation, in which we'll be splitting this DNA molecule apart, and it's easier to split 2 hydrogen bonds apart than it is 3. Keep that in mind. So we'll circle back to that at a later date, but I just want to point that out. Now, these nucleic acids in DNA are formed by anti-parallel strands, meaning they're basically head to tail. So you'll hear that called the 5' to 3' direction, or the 3' to 5' direction. Okay, it's like saying east to west or west to east. All right, so it's a directionality. And what does that mean? Here on this diagram, you can say they're drawing a line for the anti-parallel strands that say this is the 5' to 3' direction. And over here, they're showing you the 5' to 3' direction going that way. All right, so what does that really mean? I'm going to add in a slide right here. If I can figure out how to do that. So when we're talking about nucleotides, remember that the nucleotide is made up of a sugar, right? So 5 carbon sugar, here's a carbon, here's a carbon, here's a carbon, here's a carbon, and then we'll have a carbon out here. Now, if we're talking about a RNA molecule, okay, and I'm not going to draw in all the hydroxyls and the hydrogen groups, but if we're talking about an RNA molecule, all right, you're going to have an OH group here, and you're going to have one here, right? If we're a deoxyribose, okay, deoxy means that we've taken one of these hydroxyl groups away, okay? So that's the difference. One of the differences between DNA and RNA is the sugar molecule. All right, so let's go back though. Now, chemists number the carbons in a molecule. So this is carbon 1, this is carbon 2, 3, 4, and 5. You'll always find in a nucleotide the nitrogenous base associated with carbon 1. Okay, that's where it will be attached. Now, that leaves us some of these other carbons for bonding. So what you're going to have over here, remember that these DNA strands also have a phosphate group. So we have OP, double bond O right there. So there's a phosphate group. Okay, so we have a 5' phosphate group, right, at the C5 carbon, and we have at the C3 carbon a hydroxyl group, okay, and these become very important. They give us our directionality, and they are what links this nucleotide along the chain of nucleotides, right? So whenever we have the addition of another nucleotide, right, the 5' phosphate group, which is going to be here, right? So we've got 5' phosphate. I'll put it down here. And let me draw it the same way. Okay, so we got phosphate. Okay, and it's going to be attached to a 5' carbon of the next nucleotide. Okay, there's our C1. There's our 3' hydroxyl group. C2, C3. The next nitrogenous base is going to be here. Of course, these are going to be base paired, all right, to a nucleotide that's across from them in the chain, all right. So what will happen is this 5' end containing the phosphate group will undergo a dehydration synthesis with the hydroxyl from the 3' end of this nucleotide, all right. And so during this dehydration synthesis process, these are joined to form a covalent bond, right? And now we have these nucleotides are in a specific orientation. So this is the 5' to 3' direction. So we go 5', 3', 5', 3'. The next would be 5', 3'. All right, so we're 5' to 3'. All right, and if we're looking at the other strand, it would be flipped over and inverted, right? So this would be its 5' to 3' direction. Okay. And so this is the only way in which this pairing can occur. All right, but I just wanted you guys to know and realize what anti-parallel meant. Now, these DNA strands are also supercoiled. All right, they form a helix, so you can see the helical shape of this molecule. There's about 10 nucleotides per turn, even though it's not drawn here. But in addition to that, even though they're wound up, okay, there's also another hierarchical level of twisting that's called supercoiling, right? So they're twisted in this configuration, and then in eukaryotes wrapped around histones, right? But not in prokaryotes. Okay. So if I were to ask you guys, which of the following is an example of correct base pairing in DNA, which would you say is correct? I'll let you take a look at this. Okay. If you said C, you would be correct. Okay. A pairs with T and C pairs with G, and we see that correct pairing in all of those. Right? Now notice when we write as geneticists, we always write sequences, right, in the 5' to 3' direction. Okay, or we read sequences. So this sequence would be read 5' A, C, T, G, G, A, 3' end. All right, how would we read the bottom strand? We would read the bottom strand in this direction. No matter how it's laid out on the page, it reads T, C, C, A, T, GT. Okay? So remember, you always read DNA sequences from the 5' to the 3' direction. Okay, and remember the 5' end is going to have the phosphate group, and the 3' end has the hydroxyl group. Okay. Nucleotides are always going to be added to a growing chain at this end, the 3' end. So when we're adding on, when we're replicating DNA or when we're making RNA, we're always adding on to the 3' end. This is the end that grows. And we'll come back to that. All right. So now, what does the DNA look like in a prokaryote? Well, they have DNA like we do. It's double-stranded like we have, and it is circular. Okay, so they have a circular chromosome. Here is a bacterium on a scanning electron micrograph, right? So we're looking at it three-dimensionally. It has some sort of little bit of surface shapes that you can see. You can see the height of this DNA laid on, a solid surface. But the bacterium has been gently proteolized, and the DNA is being released from the cell, and you'll see it coming out in these nice long sort of loops and wavy structures. So that is the DNA in the cell. That's its chromosome. And it's hard to imagine that amount of DNA is packaged into that little bitty cell. But also what you'll see are these things called plasmids. And plasmids, like this one here, are small extra chromosomal DNAs. Okay, they usually contain non-essential genes, but we'll circle back to those in a little bit too. Let's focus on the chromosome for now. Okay, so bacteria are typically going to have one chromosome, one circular DNA. There are a few exceptions out there. There are a few organisms that have two. There also an exception where there's been bacterium found that has a linear chromosome like we do. But like I said, this having a linear chromosome is rare. Having two chromosomes for prokaryotes is very rare. All right, they also have a nucleoid, right? No nuclear membrane, so all of this is free floating in the cytoplasmic soup, although there are some, there are boundaries. So it's not just filling up the entire cell. The only reason it's spilling out and looking like this is because we've proteolized it and digested all the proteins that are associated with it. The nucleoid actually, like I've said before, is tethered by these proteins that hold it together. They kind of make it look like a ball of yarn. All right, and then there are these things called plasmids. Now, how big is the chromosome? The chromosome is about 4 megabases, like for E. coli, and I think I've mentioned before, that's 4,000 kilobases. Okay, and 4,000 kilobases equals about, for E. coli, 4,000 genes. Okay, so that's how many genes are found there. And when they looked at the human chromosome, okay, human chromosomes are each human chromosome, and you guys know that we have 46 altogether, are about 50 to 100 megabases. Ooh, so does that mean every chromosome in a human contains, anywhere from 10 to 25 times the amount of genes? Actually, turns out it doesn't. When they looked at a human, even though we've got thousands of times, actually not thousands, hundreds of times the amount of DNA that a prokaryote does. They thought it would be 100 times more complex, have 100 times more proteins. Turns out we only have about 6,000 or so genes. I'm sorry, I don't know why I said it's 26,000 genes. Okay, so we tend even though we're multicellular and we, have neurons, we have liver cells, we have all kinds of cells in our pancreatic cells, all kinds of cells in our bodies that do different things that look very different and are much larger than these prokaryotes. And then we have hundreds of times the genetic material. We only have about 6 times the number of genes. So what's all this extra stuff? Well, eventually I'm going to talk about extra stuff, but we have a lot of what we call relic viruses that inserted into our genome, got stuck there. We have a lot of non-coding genetic material, non-coding RNAs that even though they're made, they don't code for protein. They're regulatory. We have a lot of regulatory sequences in between genes because we need to be able to turn them off and turn them on. And so, and there tends to be these things called transposable elements that get copied and are termed selfish DNAs that we find in our genome, and we're going to come back to those as well. But yeah, there's a lot of extra stuff basically in the genome of a eukaryote that we don't find in a prokaryote. Why don't we find it in a prokaryote? Well, they tend to be need to copy their DNA a lot faster than we do. Okay, that's the belief, in order to compete in their environment for nutrients and space, and so they really don't need to copy extra material. It just slows them down, right? And so they limit the size of their genomes, okay, or these parameters of competition limit the size of their genome. All right, so I'm going to stop there.